So last class, we finished up talking about bone formation and bone um, resorption. And now we're going to continue on and get down into the nitty gritty and discuss more of the microanatomy here. And we're going to talk a lot of the time we'll be spending on compact bone. We will talk about some trabecular bone here. So we're going to go into a little bit more detail, especially with the anatomy here in regards to our compact bone. So if you recall in lab class, we talked about how in compact bone, we learned about what an osteon was, and that was that structure that looked like a tree ring. So you had that dark center hole with these concentric rings that went around it, much like I'm sure that most of you have probably seen a lot of trees that have been cut down and on the side of the road, and you'll notice how these trees will have, if you take a closer look at the trunks, there's rings, right, that start from the center and work their way out. And so your bones have structures that are similar to them called osteons. And they're these tiny cylindrical structures that we'll see, they make up what we refer to as the functional unit or structural unit of your bone. Okay, so that plays two roles: functional meaning physiology, structural meaning anatomy. And so we're going to see that these osteons are going to run up and down. All right, if you're looking at the bone's diathesis there, they're going to be pretty much parallel. I say a tree ring, your book likes to describe it as a bullseye. But point being is it's going to be a cylindrical structure that plays some several roles here, which we'll discuss as we kind of move through this chapter. So the first one here, the central canal, that's that hole in the center of the osteon. I'll show you a picture of it in a moment. We've seen it before in lab. That central canal is going to be where our blood vessels and nerves are located. Remember last class, we talked about how your bone is highly vascularized, which means there's blood vessels that run all throughout the bone, and it's innervated, meaning it's going to have nerves, and mainly those nerves are going to be sensory nerves because they're going to monitor and detect if there's any changes in the bone density and the bone structure there. And then out from that central canal, you're going to have these rings, which we call lamellae. And this specific type of lamella, we call them concentric lamellae. So if I show you a picture here, you can kind of see it. This is an electron microscope right here that you're seeing at the top in the middle here. All right, so you can see there's the central canal, and then you'll have these rings. You can see it nicely on this electron microscope. You can see these rings growing out from the center. And so they go around the central canal. Therefore, we call them concentric lamellae. And one of the cool things that I like about how we're designed is that your concentric lamellae, right? Each ring, the collagen fibers are angled at 45 degrees. So 45 degrees is like this. That's 45-degree angle. Now, when you compare it to the next concentric ring, you're going to see that the collagen fibers are also going to be at 45 degrees. So when you compare the collagen fibers from one concentric ring to another, they're going to be at 90 degrees from each other. And I'll show you a drawing here. There you go. So if we zoom in, here's what I'm talking about. Here's one concentric ring. You see how the collagen fibers are at a 45-degree angle? When you compare it to the next concentric lamella here, the next ring, you'll see that those are going at 45 degrees in the opposite direction. So when you compare the orientation of these collagen fibers to these collagen fibers, it's at 90 degrees. It's just one of nature's cool facts or whatnot. And so the collagen, as you recall, is part of the organic matrix. And the collagen is going to help, right, give your bone that tensile strength. Right? Collagen is one of the 3 types of protein fibers. They're long cable-like structures. And so they're going to add to the tensile strength of your bone here. And so obviously when your bone is undergoing certain stresses, it helps to resist those stresses, helps to prevent the bone from snapping, all right, like a tree blowing in the wind, all right. It will, if too much force obviously is applied to the bones, it'll snap, but it's these collagen fibers that help prevent that. All right, in these osteons, you have a cell type, and those are the osteocytes. And so they are housed in the lacuna spaces there. And it's these osteocytes that maintain the bone matrix. They're constantly monitoring if there's any changes, right, to your bone. And if there is an issue with the bone, like say part of the bone was damaged, right, or the bone matrix was damaged, these cells will help repair that. So if you recall, the osteocytes were once osteoblasts that were building the bone matrix, and they got trapped. And so they have no choice then, once they're trapped, okay, then they convert into an osteocyte. And they'll live there in the bone matrix and just maintain the bone matrix. So you'll have all these lacuna scattered throughout these osteons. And you'll notice when you look at these osteons close up, you'll see these on the microscope, they'll look like these black lines that go from one lacuna to another. And there's usually more than one. There's multiple, all right, little black lines that connect lacunas to one another. And those are called the cannuliculi. And so when you look at the shape of the osteocyte, I don't know if I have a picture here. There it is. See the osteocyte cell here? Okay, you've got, here's the body of the cell, and then it's got these appendages or these processes coming out. These processes reside in the cannuliculi. And so this osteocyte in this particular lacuna can communicate to a neighboring osteocyte in an adjacent lacuna with these projections, with these processes there. And it's through those processes where these cells can share nutrients with one another. They can exchange wastes with one another. So it's a nice little system. It's like a bucket brigade back way before we had fire hydrants all over the place in these cities here, right? You would have people line up and they'd pass one bucket to the next person, to the next person, to the next person, and then they'd throw the water on the fire, and then they'd pass the bucket back and they'd get another full bucket of water. And so that's essentially how these cells are able to transmit nutrients to one another and then transmit wastes to one another, because they don't have a blood supply, because the blood supply is in the central canal. But that's great for the osteocytes that are close by the central canal. But what about the osteocytes that are 4, 5, 6 concentric lamella removed from the central canal? And so this is one of the ways that we're able to give nutrients and wastes to the cells, so we can exchange these items, even oxygen, carbon dioxide, all sorts of things. And so the cannuliculi, these spaces allow for these processes of these osteocytes to communicate with one another. All right. So again, I've already showed you this one. You can kind of see here's the central canal, and then you can see all these concentric lacuna or lamella, excuse me, surrounding that central canal, and then throughout, scattered about, you'll see all those various lacuna, and with the osteocytes inside of them. And then you can see these dark lines going from one lacuna to the next. And so those are the cannuliculi. And so it allows these osteocytes to communicate with one another. All right. So when we're looking now at in our long bones here, we're going to talk about some structures that aren't a part of the osteon. Everything that we've talked about just a few moments ago are part of the osteon, that ring-like structure. Well, we're going to see some things outside of the osteon. And the first one here are what we call Volkmann canals or perforating canals. So the central canals go up and down, or they're parallel to the diaphysis, which is the shaft of the bone. So they go up and down. Well, these perforating canals go perpendicular, side to side. So you have all of these different canals going up and down and side to side. And so they'll connect central canals that aren't directly above or below each other to the adjacent or adjoining canal. So it makes this nice little network or this grid-like structure. Again, in these perforating or these Volkmann canals, you're going to have more nerves and blood vessels there. Again, they'll just allow communication, and you can kind of see a nice example here on the next slide. I'll come back to that previous slide there. You can see it here. So here are the central canals, blood vessels and nerves going perpendicular to the diaphysis, and then you've got your perforating or Volkmann's canals that are going to connect adjoining, all right, or adjacent central canals. So you can see that these bones have quite a bit of communication networking structures all over the place, all over the place. All right, so another structure that's outside of an osteon is what we call the circumferential lamellae. What is the circumference of a circle? Well, it's the outside of the circle, right? You know, if I draw a circle here, all right, that circle, that red line that I'm drawing here, that's the circumference. That's the outside here. So circumferential lamellae are going to be, right, these lamellae that you're going to see on the outside of the bone and on the innermost portion of the bone. So the external circumferential lamellae, these guys will sit just underneath the periosteum. And then the internal circumferential lamellae are going to sit just on the inside of the endosteum. And just to review, the periosteum is the covering on the outside of the bone, and the endosteum is that covering on the inside of the bone. It kind of lines the medullary cavity there. So let me pop over to this next slide. I'll be right back. BRB. All right, here's the external circumferential lamellae. So they go around the entire circumference of the external portion, the outermost portion of the diaphysis of the bone. Then if you look on the inside here, right, we're going to see the medullary cavity over here, but we have our trabeculae here from the spongy bone. And then you can see here on the inside, this is the internal circumferential lamellae. And so that goes on the entirety of the inside or the internalmost portion. It's just internal, or, well, I guess it would be deep to the endosteum there. And then finally, we have our interstitial lamellae. And so these are partial lamellae. So again, you are partial osteons. You'll see these osteons, and I'll show you on the next slide here. You'll see what looks like an osteon, that part of an osteon just disappeared. So you'll have in a lot of the compact bone, you'll see completely formed osteons, and then you'll notice in between these completely formed osteons, these partially formed osteons. And so these partially formed osteons are what we call the interstitial lamellae here. So if we look on our slide here, so here you can see, here are these full-on osteons. All right, but then you'll notice in between some of these osteons, you'll see some lamellae that are in existence here. And so those were from a previous osteon that just got resorbed. And so those are the interstitial lamellae in between, right, all of these other osteons here and their lamellae. Again, we see a nice picture here of the periosteum and the two layers here. You can see the fibrous, right, that dense irregular connective tissue on the outside. And then just deep to that is our cellular layer. And we'll have the various cells, right, that are responsible for bone formation and bone resorption. Osteoblasts, osteocytes, and then, not osteocytes, osteoclasts and the osteoprogenitor cells. And then again, you can see these Sharpie's fibers or these perforating fibers. And these are these collagen protein fibers that hold the periosteum onto the external circumferential lamellae there. So who would have thought that bone was this complicated? All right. Again, it's an organ. So you're going to see a lot of various structures in this organ and various tissues in this organ here. All right, so kind of zooming in again to give you an idea of these osteons, the central portion here with the blood vessels and the nerve, and then you'll see the innermost or lamellae here, right? Those concentric lamellae. As we start to separate them by layer, you can see it almost looks like a wedding cake here. Just keep in mind the collagen fiber orientation to one another. And then when we're looking inside of the lacuna here, you'll notice here's your osteocyte hanging out with its projections or its processes coming off of the main cell body, connecting it to other osteocytes through the canaliculi there. Okay, so that's compact bone. So let's look at some of the microscopic anatomy of our trabeculae or spongy bone. So a couple things. Now, spongy bone doesn't have as much to talk about as compact bone does. All right, but what you will notice about spongy bone is it has these structures, which we call trabeculae, and they're kind of like these rod-like structures, right? And it gives it that spongy appearance there. And so you'll have the trabeculae and then the pores or those openings all over the place. And that's where you're going to find your bone marrow is usually going to be in those open spaces there. So essentially, when we're looking here at the trabeculae, it's this big meshwork, what we call these crisscrossing bars. And that helps strengthen the bone because it adds resistance. It's like, I'm not an engineer or anything, but it helps with these cross braces and multi-directional force application to that tissue there to prevent it, right, from degenerating, from preventing it from injury and whatnot. So what we'll see here in the spongy bone is we're going to see, right, more lamellae, but we call these parallel lamellae. And so essentially what you'll see, and it's similar to our compact bone, but if you look here, I'll come back to that slide. Here you can see, right, here's one of our trabeculae. So all we're going to do is we're just going to cut it open, and we can see you have the parallel lamellae here, right? Almost like, but what are we missing at the center? No central canal. So you don't have a central canal here, right? But you will have these lamellae that are going to be found throughout the trabeculae. And then on the outside of the trabeculae, that's where you're going to find your endosteum. And the endosteum is going to just be made up of a cell layer. So you'll have osteoprogenitor cells, you'll have osteoblasts, and you'll have osteoclasts there. In fact, on this picture here, there's a nice osteoclast hanging out. And it looks like it's a resorbing part of the bone there. So you can see all throughout here, you'll have, these are those trabeculae there, those crisscrossing bars that run all through that give it kind of like that lattice work there. And then we'll have on the inside here, those parallel lamellae running next to each other. And then you'll notice all these little holes there. And those are the canaliculi from the osteocytes that got trapped in the bone matrix there. All right. Questions, thoughts? I know, no, it's not very exciting stuff. I promise you, we're going to get to the good stuff here. We're going to get to the good stuff. Okay, and speaking of the good stuff, here we are. So in order to really understand the ossification process, that's the process of our bones actually turning into the hardened bone structure that we're going to be talking about here shortly, we need to review, right, some of the other tissues that are a part of this process. And so we said there's other tissues in, right, other connective tissue that is going to be found in your skeletal system. And so one of those tissues is cartilage. And so we said we have two types of cartilage, hyaline cartilage and fibrocartilage. And so the hyaline cartilage here is going to play a big role because, right, I've said this before, when you start to take on that humanoid appearance as you're developing as a fetus, right, that first skeleton that you have is a cartilage skeleton. And then that's going to change over to a bony skeleton. So we're going to discuss here and just go over a few points here with the hyaline cartilage here. All right. So just to review, hyaline cartilage is supportive connective tissue or supporting connective tissue. So that means it's going to be made up of the three things that we find in connective tissue: cells, ground substance, and protein fibers. Okay. And those ground substance and those protein fibers are going to be the extracellular matrix. And so that's what we're going to see here. So when we talk about the structure of our hyaline cartilage, that's exactly what we're going to see. We're going to see the chondrocytes and chondroblasts that are going to be found throughout the cartilage matrix. And the majority of that cartilage matrix is going to be, right, protein fibers there. Right, so you have to keep something in mind when we're looking at the extracellular matrix, which is predominantly going to be our collagen. We can't forget about our ground substance. And I told you before, ground substance is essentially just these large molecules with a lot of water involved, right? And so they can be various types of large molecules. In our hyaline cartilage, we're going to see these proteoglycan molecules. But here's the important thing, and I make a big point of this. We're not going to see calcium in that matrix. I'm going to put this thought into your head. If you put calcium into that matrix, it's going to harden the matrix, right? So calcium, when it gets involved in this process, it's going to turn the cartilage into bone, right? Not exactly like that, but I want you to think, once we add cartilage to this material, to this ground substance, to this cartilage matrix, it's going to cause that cartilage matrix to harden. So we don't want to have calcium yet, right? There's a time and a place for calcium. It's just not right now, right? So some of the characteristics of cartilage is it's more flexible than bone, but it's tough like bone. It's strong. It's not as strong as bone, all right, but it's more flexible. And what we'll really see is there's a much higher percentage of water in the cartilage compared to bone. Not to say there's not any water in bone, there is, okay? But we're going to see a much higher percentage in our cartilage. And because of all these properties, that's what makes cartilage a really good shock absorber. We see that with fibrocartilage especially, but our hyaline cartilage does play a role because we said that hyaline cartilage is found, all right, on the epiphysis or on the ends of your long bones. And yes, they help to reduce the friction there in the joint, but they also do act as a shock absorber. So one of the characteristics of mature cartilage, and I mentioned this before, is that it's avascular. Now, young cartilage, developing cartilage, will have, all right, blood vessels, but mature cartilage is going to be avascular and also no nerves. So technically, if you're damaging, all right, mature cartilage, you should not technically feel that as pain because there's no nerves there. You might feel pain in other structures. That's why a lot of times when people get a disc herniation, okay, it's not the actual cartilage from the disc that's causing the pain, right? It's because the nucleus pulposus, which is the jelly at the center of the disc, is pressing on another tissue, and that tissue now is pain sensitive and it's reporting pain. All right, but keep that in mind. Mature cartilage, avascular, and it does not have nerves there. So let's review some of the cells here that are going to be involved. And you already know one of the cells, we learned about it in lab, are the chondrocytes. So the chondrocytes are going to be the cells, much like what we saw with the osteocytes, right? How their story evolved. Similar story here with our cartilage cells. So we start off with the chondroblasts, and a blast likes to build things. So the chondroblasts build the cartilage matrix. Well, during this process, those cells get trapped in the cartilage matrix, and so they get cut off. So, like that painter that paints himself into the corner, right? The chondroblasts get stuck in the matrix, and then they convert into a chondrocyte. And then those chondrocytes' job is to maintain the cartilage matrix there. And so they live in a space much like the osteocytes, called the lacuna. Okay, so there's some similarities between those cells there. And then, much like bone, some of our cartilage types have a covering of their own. Okay, bone had periosteum, cartilage has perichondrium. And there's only 2 out of the 3 types of cartilage have the perichondrium. Fibrocartilage does not, right? Hyaline and elastic cartilage do have a perichondrium there. And it plays a big role in the shape of that cartilage. And we'll see some of the other roles here as we move through this material here. All right, so that's the perichondrium. That's the covering to the cartilage. So I like this next slide because it shows you some of the similarities and differences, right, between bone connective tissue and hyaline cartilage connective tissue. All right, so you have cells that form the matrix, and they both have blasts attached to their name. So and the first part tells you what type of tissue we're talking about. So osteoblasts are in bone, chondroblasts are in the cartilage. And then as they mature, because they got stuck in the matrix, the osteoblasts turn into osteocytes, the chondroblasts turn into chondrocytes. But notice, we see calcium in the bone matrix, but no calcium in the cartilage matrix. If calcium gets into the cartilage matrix, and we're going to see this process play out here soon, it's going to cause calcification, hardening of the cartilage matrix. And that can eventually then destroy that tissue. And I'll explain that as we move on. All right. Bone connective tissue, lots of blood vessels. Okay. Highly vascularized. Mature hyaline cartilage is avascular. Avascular. Okay. So the next concept that I want to talk to you all about is how do our supporting connective tissues grow? So we're going to talk about bone growth here shortly, but we're going to start off here with cartilage growth. And so there's 2 types, but your cartilage starts to grow when you're still developing inside your mom. And so there's 2 ways that cartilage can grow. It can grow in length and it can grow in width. So you need to know these terms here. If we're talking about our cartilage growing in length, we call that interstitial growth. And that's like a tree, right? As it's growing taller and taller and taller, right? So that's interstitial growth. The other type of growth is when it grows in width, and that's called appositional growth. And so appositional growth is same thing. If we're looking at a tree, you'll notice that the trunk of a tree, right, when it's freshly planted, come back in 20 years, the trunk of the tree has gotten much thicker, right? It's grown wider, and that's appositional growth. So interstitial is when we increase in the length, it gets taller. Appositional is when it gets thicker, right? So I like to use the tree as an analogy for that. So let's talk about the steps, right? We're going to go through both types of growth here. So I'm going to start off with interstitial growth. So it's a 4-step process, a 4-step process. So what I'm going to do is I'm going to toggle back and forth. I got 2 slides here that I'm going to go back and forth. We're going to talk about the steps to the different types of growths. So the first one here, the first step that you see for interstitial growth is, right, a chondrocyte that's minding its own business, living in the lacuna, is going to divide. And notice we say it mitotically divides, right? It's going to undergo mitosis. There's 2 types of cell reproduction. There's mitosis and meiosis. What is specific to meiosis? I've mentioned this before. Right. Yeah, exactly. Meiosis only happens in the gametes, in the sex cells, the sperm, and then the oocytes, the eggs, right? Every other cell, what we call somatic cells, they'll divide mitotically, which means you'll have 1 chondrocyte, and when it divides at the end of it, right, you'll have 2 daughter cells, which will be the same as the parent cell. So you start off with 1 chondrocyte, you wind up with 2 chondrocytes, and that's what happens. So that's step 1. So if we look at the picture here on this first slide, okay, interstitial growth, and I should have told you this before. I know it was up there on the slide. I didn't mention it. Interstitial growth happens on the outside there. Or I'm sorry, internally. Right? So here you can see in step one, the chondrocyte here decides, and here it is living in the lacuna, surrounded by the cartilage matrix that it's been maintaining. It decides to mitotically divide, and it splits. What I tell folks is it's like living in a 400 feet's not too bad. It's like living in a 200 square foot studio apartment by yourself. It's fine, right? Might not be the best, but it's fine. Now try to put two people in there, right? It gets crowded. So what's gonna happen? Right now you have two cells living in the same lacuna. Well, they don't want to be living in the same lacuna. They want to live in their own lacuna. So when these cells divide, now instead of having one chondrocyte, now we've split and we have two chondroblasts. That's what we see here. All right, that's what we see here with our first step of the interstitial growth. Now, our two new cells, which are now chondroblasts, and what do blast cells like to do? Build. They like to make stuff. Yeah, and that's exactly what these two cells are going to do. So these two chondroblasts that are living in the same lacuna want to have their own studio apartment, so they're going to start producing their own cartilage matrix there. And that's exactly what they do. They start secreting this material all around themselves. And so as they start to do that, you can see, right, here they are. They're making that brand new cartilage matrix. And as they do, they're going to start to push themselves away from each other. Right? And by doing so, it's going to expand, all right, that tissue. It's going to make it bigger from the inside. So it's growing kind of like from the inside outwards. All right, so that's what we're seeing here. The chondroblasts make that new material. They push away from each other. Once they've gotten away from each other, now they're considered a chondrocyte. They're living in their own lacuna. And then what do? They basically are going to maintain, right, that cartilage matrix that they just built, or that they just secreted. So interstitial growth is not really too exciting when we're talking about, all right, cartilage. That's easy. We get to the bone, it's going to be a different story, all right? So cartilage interstitial growth, relatively easy. Let's talk about appositional growth, because remember, there's two types of growth that we're going to see. The appositional, much simpler, I shouldn't say much simpler, but similar. It's only 3 steps, but appositional growth happens on the outside, inside of the just deep to the perichondrium. All right, so when we look at appositional growth, same type of scenario. We're going to have a cell that is going to divide. So that chondrocyte divides, right? And when it does, we have 2 new cells, which are called chondroblasts. So I'm going to come right back to the slide here. Here you can see our first step. So here's the perichondrium. That's that outer covering to the cartilage. So just deep to that. So we'll see on the edge here, the outer edge, here's what's happening. You get these cells that start to divide, and they're going to do the same thing. So they've divided. Now you have 2 cells occupying the same space. They don't want to be close to each other. So they're going to start to grow and make new cartilage matrix. And again, that starts to push the cells away. At the same time, it pushes those cells that just divided away from the periphery or from the outside of that cartilage. And so it causes that cartilage to now push outwards. And the same type of scenario happens. Once they've separated from themselves, once they've gained separation from each other, and they've established their own lacuna, they'll produce their cartilage matrix until they stop. And then at that point, those chondrocytes are just going to maintain the cartilage matrix. So it's pretty much, all right, very similar. It's just basically the location of where these cells are. If it's interstitial, it's more internal. And if it's appositional, it's more on the periphery. But the same scenario is we're going to see. You're going to see a cell that mitotically divides. Then we have two new cells that are chondroblasts. They're going to grow the new cartilage matrix. It pushes them away from each other. They'll develop their own lacuna. All right. And then when they're done, they're just going to maintain that brand new matrix to which they just created. And so that's what we see here on these bottom two slides. So here they are, still sharing the same lacuna, but they're growing that matrix and they're pushing, all right, away from the periphery. And you can see they're moving deeper down into that cartilage. And now they're two separate cells, right? At that point, they're two separate chondrocytes, and they're just going to live in their own lacuna until possibly if they divide again. And the same thing will happen. Okay. So again, like I said before, we're going to see this process of cartilage growth occur when you are in your early stages of development inside a mom. Right? It's actually, believe it or not, this is going to happen, all right, earlier than 10 weeks. It's pretty soon, right? And as so, all right, as this cartilage, once you've gotten to that point where you've kind of stopped the growing, you know, if you ever really do stop growing during the fetal development, but you'll start to see that the cartilage growth will kind of back off a little bit. Interstitial will happen quite quickly. All right, quite quickly there. So what will happen is once this process is completed, now when we have gained our fully mature cartilage here, we won't see any new cartilage except here. Right, when you hurt yourself. Because like most tissues, then you'll repair yourself. Has anyone ever heard of glucosamine chondroitin sulfate? If you've worked with an orthopedist, maybe, especially if they're folks that deal with knee issues. So glucosamine chondroitin sulfate is essentially the ingredients for ground substance. And so what happens is you take this material, you're providing your body with ground substance to help repair damaged cartilage. So a lot of times, doctors, healthcare specialists will tell patients, especially if they're getting degeneration of joints, all right, called DJD, degenerative joint disease. One of the early ways of intervention for that is to tell people, hey, you've got some damaged tissue there, some damaged cartilage, take this glucosamine chondroitin sulfate, and that will help repair the damaged tissue. It might not necessarily make it where you'll regrow the damaged cartilage, but it helps slow the process down, and it also helps to repair some of the existing material there. But you don't really see it regrow new cartilage. Unfortunately, as we get older, your body just doesn't do that. And the reason why is no blood vessels. No blood vessels. That's a problem. Okay. Okay. So now this leads me into the next process here, right? And that is the process of bone formation. And we give it a name. We call it ossification or osteogenesis. Osteo being bone, genesis is creation or making, right? So ossification is when we are going to now form bone tissue, right? And so this starts to occur anywhere between the 8th and 12th week of development. You might see that on a test. Okay, 8 to 12 weeks. That's early, right? That's first trimester stuff. So you're going to start growing bone then. And the even crazier part is prior to the ossification is that you already have kind of a humanoid skeleton, except it's just made out of cartilage. Made out of cartilage. And so this ossification process is going to occur, all right, starting at the 8th to 12th week, and it's going to continue on through your childhood, through your teenage years of adolescence, and possibly into early adulthood. So this goes on for a while. So again, we're going to now talk about there's two ways for ossification to occur. You can get it what we call intramembranous ossification and then endochondral ossification. And so those are going to be a little bit similar to what we just talked about for the cartilage for the interstitial and the appositional growth there. All right, so intramembranous and endochondral. If you dissect those words there, intra means within, right, a membrane of some sort. And then endochondral, chondral has to do with cartilage. We're going to be dealing with cartilage during this point here. All right, so let's start off with the intramembranous ossification. Here again, like I said, we're going to grow the bone inside of a membrane here, and you can see all the different places as this occurs. Your cranial or flat bones of the skull and some of the facial bones there, your very uniquely shaped mandible, and for whatever reason, don't ask me why, all right, the middle part of your clavicle. So your clavicle, which we now know is the collarbone, because we learned that earlier this week, right? The clavicle is going to be one of the bones that has both intramembranous ossification and endochondral ossification. All right, so basically, as I go through the intramembranous ossification, there's fewer steps in intramembranous ossification compared to endochondral ossification. I'm going to go step by step. One step, then I'm going to show you a picture so you can kind of correlate things, right? So there's 4 steps that go on there. All right, so the first part, or step 1 for intramembranous ossification, you're going to have these clumps or this group of mesenchyme. And we all know what mesenchyme is, right? Mesenchyme is that tissue, right? That embryonic stem cell tissue. That's where all the connective tissue comes from, right? So you're going to have these clumps of mesenchyme tissue. They're going to start to differentiate and yield osteoprogenitor cells. And so, as you recall, those osteoprogenitor cells are going to do one thing. They're going to divide into an osteoprogenitor cell and yield us an osteoblast. Okay? And then some of those osteoblasts are going to start doing what osteoblasts do. Secrete osteoid, the organic component to bone. They're going to start building bone, or at least start the process of building bone. So looking at this picture here, look all the way to the left there. Here it is. Here's your mesenchyme. And so you'll start to see, all right, this grouping of mesenchyme. And so these osteoprogenitor cells will start to yield us some osteoblasts. And these osteoblasts will start to secrete osteoid. Right? And so remember what osteoid is. Protein fibers, primarily collagen, right? And then ground substance. Proteoglycans, glycosaminoglycans, right? And so those are going to help that's our wet cement. That's our wet cement. So what do we need to harden our wet cement? Calcium, nailed it. Yeah, all right. And that's why I told you, all right, calcium, when we start to invite it to the party here, it's going to start to make things harden, all right, through that process of calcification. We'll talk about that here in a second. Okay, so that's step 1. So we get these ossification centers, which is just these areas where our mesenchyme is located. Okay, so now what we're going to see in step 2 is that process of calcification. So calcium is going to infiltrate that area. These calcium salts start to infiltrate into these ossification centers here. And they're going to do what calcium salts do. They're going to deposit these calcium salts and crystals around the collagen fibers there. And then it's going to start to harden the osteoid. It's going to help our wet cement start to dry. And what happens to these poor osteoblasts that are in that hardened, right, osteoid? Well, they're going to get stuck there and they're going to convert into osteocytes. Let's just see that in a picture and I'll come right back to this. So that's what you're seeing here on our second slide picture here. Here you can see our osteoblasts secreting the osteoid. Those calcium crystals or calcium salts infiltrate the area, and they start to then precipitate out around the collagen fibers, and they start to harden the osteoid. And these poor osteoblasts get stuck in it, and they have no choice but to convert into osteocytes. And what's the job of the osteocyte? Maintain the bone matrix. Maintain. They just made it. Now they got to live in it, and so they got to maintain it. Keep their apartment clean. All right, so that's what we're going to see here in step 2. So the osteoid that was made in step 1 is now going to calcify. It's going to harden. Cool so far? Not too difficult. Okay, that moves us on to step 3. So that hardened osteoid is an early version of bone. It's not the best kind of bone, but it'll have to do. And so we call that woven bone. Right? So, like I said, it's not the best because 1, it's brand new, and 2, it's not really well organized yet. We haven't gotten our osteon formation going on and all that fun stuff. Right? So, what we're going to see now in step 3, especially when we're near the periphery, we're going to see some of that mesenchyme now start to produce our periosteum. All right, I'll come right back. That's what we're seeing here in our third picture. All right, so you can see this process here is continuing on here, but you'll notice a few things. One is, before I talk about this, you'll see on the periphery here of our bone, our woven bone, right, that mesenchyme starts to convert into the periosteum. So we're going to start to form, all right, that dense, irregular connective tissue layer, the fibrous part of the periosteum, and then we'll get that inner cell layer that's going to start to form here. But what do you notice here? Blood vessels. Yeah. So now blood vessels have managed to infiltrate into this area. So now we're going to turn this whole process up on high heat, meaning with our blood vessels now we can bring in tons of calcium. And we can start to really provide nutrients for these cells as they're making the bone matrix, bring in more calcium, and start to harden a lot of these ossification centers, on a high kind of cycle going on here. And so that's what we're going to see here in that third step there, right? So we're going to see the formation of our periosteum, and we're going to see a lot of that woven bone being created, right? And especially with the infiltration of the blood vessels there, it really helps the process move along relatively quick. Okay, our last step is now we're going to convert that immature, poorly made, all right, poorly organized woven bone into what we call lamellar bone. And lamellar bone, this is a very similar process occurs when you get a bone fracture. Because when the bone starts to heal, you form woven bone first just to kind of help stabilize the bone. And then a lot of that woven bone gets replaced by a better type of bone, which is the lamellar bone or that secondary bone here. And so this is a similar process that you see in a bone fracture here. Because now what we're going to see is this lamellar bone is going to yield us both compact and spongy bone. And especially, all right, with that spongy bone, we're going to get the trabecular formation occurring there. All right, because keep in mind, this process, this intramembranous process, right, primarily, you saw what bones it occurs in. Right? And we talked about the cranial bones, those flat bones there. Those types of bones don't have a medullary cavity. So remember that I was talking about the sandwich analogy here. It's got 2 slices of bread, right? That's the compact bone, and then the filling in between, that's going to be the spongy bone. And that's what we'll see here, right? With that lamellar bone there, we're going to start to get these 2 layers on the outside of compact bone, and then that nice, creamy, spongy bone filling in between, right, our 2 compact bone layers there. And that's what we see here in that last picture. Right there. So now we have our compact external layers here and here, both the top and bottom. All right, we have that nice periosteum covering on both external surfaces here. And then you'll notice then internally in the center, we have the trabecular bone there. Again, with the pores there, the holes inside the trabecular bone with the trabeculae. I keep saying trabecular bone, spongy bone, same thing. All right, you get the spongy bone with all these trabeculae all over the place. So that's how we form, all right, our a lot of our flat bones and some of the facial bones in the central mandible, central part of the clavicle. Now we're going to get to the other type of bone growth. And so there's several more steps in what we call endochondral ossification. But without further ado, let's just jump right into that. Okay, so endochondral ossification. The nice thing is, with this type of bone growth, we start off with an already existing model. But what we're going to do is we're just going to I won't say pull the model apart. We're just going to replace a lot of the tissue in that model with bone. All right, but there's several steps to it, okay? Several steps, like 6 or 7 steps. All right, so we're going to first start off with our model, which of course is going to be made out of hyaline. And so a lot of the bones found in your skeleton are going to engage in this type of ossification here. So we're going to use long bone example, and we're going to go back and use the humerus specifically for this. That's what your book uses, so that's what I'm going to use. Make it easy on you. All right, so there's going to be several steps, and they're a little bit more detailed. And again, I'm going to do the same thing. I'm going to cover one step and show you the picture, and then we'll move on. All right, so let's start off with step 1, which is easy. All right, so the fetus is going to make that hyaline bone model. Okay, know the dates, 8th through the 12th week. All right, 8th through the 12th week. So the cells that are involved in the cartilage matrix, right, you're going to have the chondroblast producing the matrix, and then they're going to differentiate into chondrocytes, and they'll maintain the matrix. So I'll show you a picture. I'll come back to that slide. Here you can see, easy. This is the easiest step out of all of them. There it is. Right? 8 to 12 weeks, baby has that humanoid appearance there, and we start to grow, all right, well, we don't start to grow, but we are going to start that ossification process. But it starts off with this bone here being completely made of hyaline cartilage, right? And it's got its own perichondrium covering that. Right, so that's step 1, easy peasy, right? Okay, so now we're going to move on to step 2. Now we're going to replace some of this cartilage material or tissue, and we're going to start to grow our bone tissue here. So here's what happens. And remember, I told you, we don't want calcium in our cartilage because this process will happen. And once it happens, we can't really go back. So your cartilage is now going to undergo calcification. And so here's what's going to happen with calcification. All right, so your chondrocytes that are trapped inside the matrix, right? Once calcium infiltrates into that calcium matrix, it starts to cause that calcium matrix to harden. And when it does that, those cells are cut off from getting any type of nutrients and getting rid of any type of cell waste. So, has anyone ever heard of a game called SimCity? All right. So one of the early, I don't know what made me think of this, but I had to say it because I was going to ask you, what would happen to you if we put you in a closet and locked you in that closet and you couldn't get out and we left you there? What would happen? Well, you die, right? If you could never get out and you were stuck there. That's what happens to the cartilage. That made me think of SimCity because for whatever reason, I don't remember which version of it, but if you put somebody in a closet, they would die and turn into a zombie. Y'all know that? And the same thing would happen if you put them in the swimming pool and took the ladder out of the pool. Anyways, I don't know what made me think of that. But anyways, so that's what's going to happen here, right? So the chondrocyte is going to be stuck in this hardened matrix. It cannot get nutrients. It can't get rid of its waste. So it starts to die. And when it does, it decomposes and it leaves a hole. So now in all this calcified cartilage matrix, you have all of these holes all over the place. And so in some places, you'll have more cells kind of in a group, so you'll get larger holes here. Well, here's what happens. Now you start to develop these holes, and blood vessels are going to infiltrate into that tissue and start to grow into these holes there. And when that happens, we're going to start to bring in, all right, our special bone cells, our osteoblasts. So the bone cells kind of ride on in, okay, through those blood vessels there and other means there. And so they're going to do what they do. And what is that? Build bone. So now they're going to infiltrate that area around where that dead cartilage matrix is existing. And they're going to start secreting their osteoid. And then what happens if we've already got calcium that's already infiltrated that area? Well, the calcium then deposits into the osteoid. And what happens? Those calcium salts then cause calcification, that hardening of the osteoid there. And so this first happens in the diaphysis. I'll show you a picture. So that's what we're seeing here, right? During, again, fetal development here, still haven't been born yet, right? But again, this is we're going to start to see this, all right, weeks 8 through 12. So you can see here are the cartilage cells. Calcium has infiltrated the area. It's caused calcification and hardening of the calcium matrix. The cells start to die. They start to produce these holes here. Blood vessels infiltrate, start to grow into that area, and they'll start to bring in the osteoblasts and more calcium, and it just starts to spread all over that area there and lay down osteoid over the old, dead, existing calcium matrix there. Okay? And so it goes in there and it starts to replace the calcium tissue, the cartilage tissue with bone tissue. And it first happens down here. I say down here. Here in the diaphysis. Here in the diaphysis. So you start to form bone tissue in the diaphysis. So we call that first formation of the bone tissue there the periosteal bone collar. That's what we start to see. Meanwhile, on the epiphysis, on the ends of the bone, it's still hyaline cartilage until the third step, okay? So the first area of ossification, we call that the primary ossification center. That's where we start to see ossification happening first, initially. All right, so that is stage 2, right? Or step 2, or whatever you want to call it, phase 2. Calcium dies. We bring in the osteoblasts. They start to create the osteoid, start to calcify the osteoid, and we start to form bone tissue. This happens in the diaphysis. Okay, the third step. The third step here. All right, we've started our formation here of the ossification throughout the diaphysis here. And meanwhile, keep in mind, all right, that diaphysis area of ossification, we call that the primary ossification center. So the nice thing is that calcium was very nice in regards that it kind of created that model, that template that your bone cells can now just model over. There was I don't even know if they still do it, but my mother broke her tibia. Literally, she was down here visiting my brother way before I had moved down here. And this is why I'm the favorite son, by the way. My brother's dog, who was a very high energetic boxer, had hit my mom playing in the backyard, hit her in the knee, and pretty much sheared off the top of her tibia, which is the major weight-bearing bone in your leg. Right? And so she had to have surgery down here. Well, at the time, the surgeon was using sea coral. If you've ever seen sea coral, like dried sea coral, it looks like a sponge, doesn't it? It looks porous and whatnot, all right? And so they used sea coral, sterilized, obviously, and they put it into the area of the top of her tibia, and then her bone cells pretty much remodeled the bone around the sea coral, and because it's very similar to calcified cartilage. Again, I don't know if they even still use that process, but that's basically what we're seeing here. We're seeing, all right, your bone cells use that cartilage template there, all right, to build around the cartilage and build that bone matrix there. So your primary ossification centers for the bones that are pretty much undergoing endochondral ossification have pretty much formed by the 12th week. Now, obviously, that's not like the only time it happens. It could happen as early as the 11th week, and sometimes as early as the 10th week or a little bit later. But pretty much by the 12th week, all right, the primary ossification center has occurred here. And so in the center of the bone here, all right, is where it begins. And so it goes out towards each epiphysis. So towards the proximal epiphysis, towards the distal epiphysis. And so as it does that, right, the calcified cartilage matrix has died, and then the osteocytes come in, or the osteoblasts come in, and they remodel over that. So really what we're thinking, what you're going to see here is this whole process, that bone connective tissue displaces that hardened, calcified, dead, degenerated cartilage matrix there. And what plays a big role in that? The fact that we got our capillaries that infiltrated the area, right, and the osteoblasts that have also infiltrated that area. So that's the step 3, which I kind of talked about a little bit here when we were going over step 2. So you can really see it on supercharged here. So we start here in the middle and we start to grow out towards each of the epiphysis. And now we're going to start to see, right, as the diaphysis around week 12, as it's done this, now we're going to see our secondary ossifications start to form, all right? And we're going to talk about that in step 4. Okay, so step 4 begins with the secondary ossification centers. And so these occur in the distal and proximal epiphysis. Same type of scenario, y'all. So if you understand what happened in the diaphysis, you really don't even have to worry too much about what's happening in the epiphysis, because it's the same thing, right? So hyaline cartilage calcifies, killing the cells, causes death and degeneration. And then what's going to happen is our blood vessels will infiltrate the area, along with, in this time, osteoprogenitor cells, which are pretty much going to then convert into osteoblasts. All right, and so this happens, and it continues to occur all the way up until birth. So when you're born, you're still having the formation of your secondary ossification centers. And then also, we're going to start to see the formation of the medullary cavity. So we're going to start to form that hollowed area in the middle or the center of a lot of these long bones there. All right, so what creates that medullary cavity? The third cell that we haven't talked about, the osteoclasts. Because if you recall, I'll come back to this slide. If you recall and you look at this picture here, here you can see, all right, there's no medullary cavity here. We got to hollow this thing out. This is when we bring in the osteoclasts, because that's their job, right? To resorb bone material. And so that's what they'll do. They'll start to just hollow this area out, right, in the diaphysis, because that happens first in the primary ossification center here. So when we get here to step 4, right, we're starting to get the secondary ossification centers here. Oh, you can't see that. I didn't realize that. Here I'm moving the cursor around and it's not showing up on the screen. All right, you're going to see here in the secondary ossification centers, as they're occurring back down in the diaphysis, osteoclasts have infiltrated the area and they start hollowing out and making that medullary cavity there. So this all happens in the 4th phase there. All right, I'm going to stop here for a second and ask if there's any questions. I know it's a lot. It's like a story, though, right? It's story time. If you like story time, any of you with kids, and you read stories to them, read this story to them tonight. All right, try it. Try it. I'm always trying to tell my kids stuff. They never want to listen to me. Okay, so that's step 4. We're almost done, right? We're almost done with endochondral ossification. Okay, so let's move on to 5 and 6, the last 2 stages here, or steps, or whatever you want to call them. If you notice, step 5 is really basic in the fact that it just says, okay, bone replaces cartilage. We know how that process works now, okay? And then you're going, and this is important that you understand this. We're going to replace all the cartilage in the bone except for 2 areas, which makes complete sense. We're not going to get rid of the cartilage that lines the outside of your bone on the areas of where articulation or joints are going to occur. That's called the articular cartilage. And we're not going to get rid of the cartilage in the epiphyseal growth plates. Those are the only 2 places where cartilage will stay. So we're going to keep the articular cartilage because, again, it reduces friction in joints and it helps to act as a shock absorber. So we don't want to get rid of that. And then, of course, we don't want to get rid of the cartilage in the epiphyseal growth plates because we want to grow. But we're going to get rid of it everywhere else. So that's what you're going to see. I'm going to come back to the slide. Here you can see, and this is an ongoing process. If you noticed, here, let me go back one. If you notice here, if you've been looking at these pictures as I've been going through, I know I've been focusing on the drawings, but look, fetus, newborn or baby, okay? And now we're seeing here with step 5, and this goes on for many years, all right? We're talking about a young kid. And so we're seeing as this child is growing and developing, the medullary cavity is getting bigger and bigger. Hence, one of the reasons why a lot of your long bones no longer have red bone marrow, because we're now producing that medullary cavity and we're getting rid of that space in there. And you can see we've pretty much gotten rid of most of the cartilage, except for the articular cartilage that lines the ends of the bones here, and then the cartilage that's going to be in the epiphyseal growth plates there. All right, and so this goes on for many, many years, all right, until we get to the last stage here, or last step, which is going to be where everything stops with the growth, right? When the epiphyseal growth plates eventually are done producing cartilage and ossify. And then at that point, they form what we call the epiphyseal lines there. And so this will go on. And obviously, some bones will grow longer than, no, I shouldn't say longer than, will grow for a longer period of time than others. And so we'll see, all right, the epiphyseal growth plate turn into the epiphyseal line. And that's what we'll see. I'll come back to the slide. That's what we see in this last picture here. Right there. Full grown adult. I mean, look at that bone there. How big the medullary cavity is. But look, tons of blood vessels in there. We can't see it on this drawing, but there's going to be some nerves there. No longer do you have an epiphyseal growth plate. Right? You still have articular cartilage. That's not going anywhere unless it's degenerating. But pretty much all the cartilage in this bone, except for the articular cartilage, is now gone. And you're done, and you're pretty much done growing. All right, questions, thoughts on that? If you really understand just the basic pattern, starting off with the cartilage model, cartilage gets calcified, cartilage matrix dies, there's holes all throughout, blood vessels infiltrate into those holes, osteoblasts invade for the primary ossification center, and then they just do what they do, they make bone. And then the secondary ossification center, similar type of situation, osteoprogenitor cells, infiltrate into there, same type of thing, right? So again, go over this a couple times and just to kind of nail it home, just so it makes it a little bit easier to understand here. All right, so when we're looking here at bone growth and whatnot, you have to understand something. Your bones are constantly remodeling themselves, constantly. And so this is going to be obviously what we just saw, a good example of bone remodeling with the formation of the medullary cavity. Okay, and as the bone grows longer and longer, so will that medullary cavity. So it's going to be remodeling that. So osteoclasts are going to be working on tearing down bone, while the osteoblasts are going to be working on building the bone here. So when we're looking at bone growth and remodeling, we'll see that happen during the embryological development there. And when we're talking about, keep in mind, we talked about this with the cartilage. When you grow taller, we call that interstitial growth. And so increase in the growth of the bone lengthwise is interstitial, like we said with the cartilage. And then increase in the width or the diameter of the bone, we call that appositional. So I'll start talking about this, but I won't be able to finish it. And then I'll have to send you guys out a video on this. All right. Interstitial growth here, that is the growth lengthwise, and what's responsible for that? The epiphyseal plate. Okay, so in the epiphyseal plate, on one side of the epiphyseal plate, you're going to see, right, the growth of cartilage tissue. So the chondroblasts will be producing cartilage. And then on the other part of the epiphyseal plate, what we had seen, what we just talked about with endochondral bone formation there, we're going to see that cartilage that was grown by those chondroblasts, it will calcify, it will then harden and then degenerate, and then the osteoblasts will come in and they will start to remodel over that cartilage there. And this happens, there's a growth plate at the proximal epiphysis and at the distal epiphysis. So it pushes the bone in either direction. Let me show you a picture. Here you can see, all right? So these, well, the growth plates here are going to push the bone up that way, and then it's going to push the bone down this way. So it pretty much grows from the center out, and it makes the bone longer. And that's what we talk about when we're talking about that interstitial growth there. So keep that in mind. And during that process, we're going to push that cartilage towards the epiphysis. And then eventually that cartilage that was just produced will calcify and then be converted over into bone tissue. All right, questions? I think, yep. Let me stop here because it's a good place to stop. All right, so let's take a few moments and take a break.